Generating a CAR T-cell product with functional competence that engraft, proliferate, and has cytolytic activity following infusion is a key component of an effective adoptive immunotherapy. For example, in solid tumors, CAR T-cells, they can penetrate solid tumors and they receive optimal antigen stimulation. However, their overall engraftment and proliferation is impeded in vivo. The main benefit of this approach is that you can retain the quality of the CAR T cells or differentiation state of the CAR T cells without generating terminally differentiated or exhausted T cells, which would have diminished proliferative capacity or poor effector function following infusion. This technique can be difficult because T cells are very sensitive to their cell concentration or the surface area of the vessels that they're cultured in, as well as their metabolic needs. Essentially, T cells need to be kept happy throughout the process, otherwise they won't grow.
To begin this procedure, set out 6 well-cultured dishes. Activate fresh or cryopreserved primary human T cells by mixing them with anti-CD3/CD28 magnetic beads at a ratio of 3 beads per T cell in the wells of the culture dishes. Culture the T cells in ex vivo 15 medium, supplemented with normal human AB serum, L-glutamine, HEPs, and IL-2. Activate the T cells at a concentration of 1 million T cells per milliliter during expansion, and culture them at 37 degrees Celsius with 20% oxygen, 5% carbon dioxide, and 95% humidity. After overnight stimulation, add lentiviral supernatant to the activated T cells. Calculate the volume of supernatant.